you. So today I'll be presenting stuff that I did while doing my PhD, which I did under the supervision of Jan Robert Bars in the Biocontrol Research Unit in University College Dublin in Ireland. So part of my PhD looked at using molecular approaches in biological control, and in particular using DNA barcoding to resolve taxonomic ambiguity between geographically distinct populations. So my PhD focused on pre-evaluating biocontrol agents for Lagarosiphon major, and this is a submerged aquatic plant from South Africa that's become naturalised in several European countries, Australia and New Zealand. And although the distribution in Ireland is quite limited at the moment, the need to control it has been identified because of all the usual problems associated with invasive plants. Probably that's most important for Ireland is the fact that it significantly increases the risk of flooding. So a number of different methods have been tried and tested, including chemical and mechanical, and they've had varying levels of success and suitability. And most importantly, they've totally lacked any level of sustainability. So biological control has been put forward in the hope that we'll find a more permanent control solution. So the biological control program was initiated in November 2008, and the first large-scale survey was conducted here in South Africa, and a number of agents were identified to be promising, and the most promising one was a leaf mining ephedrite fly. And we later brought this back into the quarantine in UCD, and since then it's undergone a pre-evaluation study. We've looked at the potential colonisation success of the fly in Ireland and Europe, the damage potential of the fly, and also the host specificity in an Irish context. And more recently, we've also finished host specificity for the test plant list in New Zealand. So a vital step when considering a natural enemy's potential for biological control is the ability to accurately identify it. And this is where taxonomic identification comes into play, and it allows the species to be scientifically recognised. This is really important, particularly for a newly discovered species like this fly is. So John Deeming of the National Museum of Wales described it for us, and he named it Hydrellia Lagarosiphon, and it's distinguishable from other Hydrellia species based on variation in the male genitalia. And I'm sure anybody who's had to get a species described, and you often do find these species when you're looking for natural enemies for biocontrol, it takes such a long time, and it's really, really frustrating. And it's very nice of him to do it. He came out of retirement, but it took 4 years from start to finish. So one of the problems with taxonomic identification is that you're relying on morphological features, and sometimes these are indistinguishable, and there are a number of biological control agents that are not readily identifiable based on their morphological features alone. So there are subtle differences often in their host range or their thermal tolerance, but morphologically they're exactly the same. On top of this, taxonomy is a field facing huge funding challenges, and I'm sure anyone that does need stuff identified, it's quite hard to get taxonomic experts these days. So as this has been happening, genetic barcoding has emerged as a cost-effective tool to rapidly identify a species, and this is a technique that's much more readily available to people like me who don't have taxonomic expertise. So the most widely used development in genetics is the using this mitochondrial cytochrome C oxidase subunit 1 gene to identify species, and also to look at genetic variation that exists within populations of the species. And this is what we set about doing. So we're lucky enough to get to come back to South Africa a couple of times since 2008, and using SAMBU records we targeted localities of Lagarosiphon major. And the fly that we were looking for was found throughout the geographic range surveyed, but often the sites were incredibly isolated in terms of their elevation and ecoregion. So we collected adults from over 20 sites on Lagarosiphon major and Muscoides, and in a couple of cases also on a Potamogeton species. We also collected pupae from each of these sites, and we reared the pupae to the adult stage and also used them in the analysis. We included individuals from our lab culture that was undergoing host specificity testing, and 2 other lab cultures that we imported into the quarantine facility. And we brought these in to screen potential performance differences between populations from different areas. So we're looking at their host range and their thermal tolerance. And I also included a number of outgroups, so these are other Hydrellia species, just to put the differences into perspective. So the objective of the study really was to investigate the degree of differentiation between populations from different areas and determine whether or not a subspecies or a geographically distinct lineage occurred. And then this can steer subsequent decisions on whether or not we should be testing the geographically distinct populations separately, or should we be targeting particular localities to collect agents from. So the relationship between the mitochondrial haplotypes basically created this network. So it's a bit of a mess, don't worry about that. What it really tells you is this haplotype, the large circle on the top, it was the most commonly encountered haplotype. And from this, the 40 other haplotypes evolved. And what it's showing us is that there's a very low level of differentiation. And as you can see, there's very little geographic structure. So individuals that were collected at the same sites didn't necessarily group together, and individuals that were collected at different sites did in some cases. We also generated a phylogenetic tree to ascertain the relationship between the different individuals. And the majority of the specimens we collected during the survey grouped into this clade here on the top. But again, you see that there's no discernible geographic pattern within that clade. The remaining clades represent the other Hydrellia species, which are the outgroups. And then there's this annoying clade at the bottom that causes a couple of problems. So just look at them a little bit more closely. When we were carrying out the genetics work, instead of using the entire fly, we just pulled off a couple of the legs. And this is great because then we still had the remainder of the fly left if we ever did need to go back and look at it or get it taxonomically described. And luckily we did do that, because it turns out the Leidenburg fishery samples and the Dundee samples were actually those that are found on the Potamogeton species. So this would indicate that that fly that we found on the Potamogeton species is not Hydrellia Lagarosiphon. The Hendrina sample, on the other hand, was slightly more problematic. But what we did realise is that any of the Hendrina adult samples that we collected in the field are represented in this clade down here, but any of them that we reared from the pupae sat in the clade with the remainder of the specimens. So this highlights the importance perhaps of collecting the immature stages that are definitely associated with the host plant that you're looking for. So far the work would indicate that there's no significant geographic pattern, and when we carried out analysis of molecular variance, it gave us a negative variance. And we also know that haplotypes were found across, there were shared haplotypes across all 3 provinces. So when you've got no structure and shared haplotypes, this would suggest that there's relatively high gene flow. But I think this is incredibly unlikely considering the large distances between some of the sites, the fact that some of them are really isolated, and the Drakensberg Mountains would also act as a natural land barrier. So we looked at the genetic diversity indices for an answer. So you have high nucleotide diversity and high haplotype diversity. And when you look at the relationship between the 2 of these, it can be explained by various demographic frameworks. And basically all it's telling us is that the population is stable and has a relatively long evolutionary history. And that's why we're not seeing any geographic pattern or any genetic structure. So the data confirms that it's a single leaf mining species that damages Lagarosiphon major over a very large area in South Africa. The specimens on Lagarosiphon muscoides are also confirmed as Hydrellia Lagarosiphon, but those that were found on the Potamogeton species are not. The individuals that we used in our host specificity testing are from one population, and luckily for us there seems to be an apparent absence of a cryptic species, which just makes life so much easier. And I should also mention that the other two populations that we brought into the quarantine displayed no differences in terms of their host range or their thermal tolerance. So just some final thoughts on the molecular approach. So I think conducting population genetics prior to release does provide some really useful baseline data on population genetic variation. And I think it adds another level of confidence which can guide the safety of biological control programme. And this is really important for us in Ireland because we've never released a biological control agent before. So we have to be seen to tick all the boxes. But I will say this, genetic barcoding has proved to be a worthwhile endeavour, but it does have its limitations. And I think that it should be used to supplement rather than replace morphological data. Thank you.